Good afternoon. My name is Mikey Kostecki, and I would like to welcome you to my Spring Science Symposium talk. This year, I have conducted a research project aiming to assess the aerosolization of non-tuberculous mycobacteria in shower air and to evaluate the effectiveness of a dehumidifier in reducing NTM aerosols. Non-tuberculous mycobacteria, or NTM, are opportunistic infection-causing pathogens that can be found in natural waters, soil, and municipal water systems. NTM are a member of the Mycobacterium genus and encompass all mycobacteria other than Mycobacteria tuberculosis and Mycobacteria leprae. There are roughly 125 species of NTM, of which roughly 60 are suspected or known to cause disease. NTM pulmonary disease is the most common manifestation of an NTM infection. Though NTM is practically ubiquitous, NTM pulmonary disease is most commonly linked to predispositions and biological structural deficits. In the United States, NTM-PD prevalence increased annually at a rate of 8.2% per year from the years 1997 to 2007. Though the national prevalence of NTM diseases have increased, the regional distribution is not equally dispersed. As shown in the figure, the prevalence of NTM-PD in the Southwest and Southeast regions were disproportionately higher than that of the US Midwest. It is important to note, however, that the state with the highest prevalence of NTM-PD is actually Hawaii. Hawaii, with a prevalence rate of 396 cases per 100,000 people, had roughly 4 times the national average incidence rate. It is thus evident that regional and climate differences may have an impact in the prevalence of NTM infections. Previous studies have established a link between NTM infections and infected patients' household showers. One of the major reasons that NTM are able to enter municipal water systems is due to the fact that they are disinfectant resistant. Thus, NTM are able to survive many of the water treatments and then are able to enter houses, hospitals, and practically any water source. One defining feature of NTM is its lipid-rich cell wall, which helps it to survive in rough environments. These lipid cell walls can comprise roughly 60% of the bacterium cell mass. Studies have shown that the lipid cell wall plays a key role in the aerosolization and distribution of NTM. In one study, it was determined that the NTM attached to water bubbles in the air and burst into water droplets small enough to enter human lungs. Another study in 2018 looked at the relationship between a household humidifier and the aerosolization of NTM. Their data clearly showed that when a humidifier was ran, NTM aerosols spiked. However, when the humidifier was turned off, NTM aerosols returned to near normal levels. All the previous studies have shown the importance of better understanding the aerosolization of NTM, especially in showers, so that effective intervention protocols are created to assist at-risk individuals. Additionally, these studies reveal a need to find a way to decrease NTM aerosols that are already present in the air. Therefore, my study aimed to answer 2 questions. First, is running a shower an effective aerosolizer of NTM? Similarly to a humidifier, a shower pumps massive amounts of water droplets into the air. If water droplets can carry NTM, it seems logical that the more water droplets in the air, the more NTM aerosols there would be. Therefore, we hypothesized that a running shower would be a significant source of NTM aerosols. Secondly, we questioned whether the amount of NTM aerosols could be reduced using a dehumidifier. We hypothesized that by removing water droplets from the air, we would also decrease the NTM aerosols because they would have less surface on which to aerosolize. So to answer these questions, I modeled my study on the average human shower routine and sampled the air at 3 distinct points. On this slide is the general flowchart. So the first air sample that I did was pre-shower. I then ran the shower and sampled the air again post-shower. Following that, I decreased the humidity in the shower and sampled the air one last time as post-dehumidification. My last step was to swab the shower head. So using an impaction air sampler, which sucks in air and directly plates bioaerosols, I sampled the air pre-shower, which served as my first control. Note that the impaction sampler was placed 6 feet directly below the shower head. Following this first sampling, I ran the shower for exactly 8 minutes because the CDC cites that the national average for shower runtime is 8 minutes long. Immediately after, I resampled the air as my experimental group 1. Then, by using a household dehumidifier and opening all the windows, I decreased the relative humidity in the bathroom by 10%. Once the humidity dropped 10%, I again resampled the air as my experimental group 2. Finally, I carefully removed the shower head and using a sterile swab, I collected the shower head biofilms. To identify the bacteria present in the shower head itself, I plated the swabs using both a clean and disinfected method. The clean method was done simply by plating the contents of the swab. In the disinfected method, the contents of the swab were treated with CPC, a chemical commonly used to kill all bacteria other than NTM. Then, all plates, including both the swabs and air samples, were incubated for 17 days at 30 Celsius and 37 Celsius. On day 17, I came into the lab and processed suspected NTM colonies and prepared them for PCR and sequencing. I used both a 16S primer and RPOB primer in the PCR reactions to replicate specific strands of DNA that would help me identify if an NTM was present. Positive RPOB samples were then sent to Large & Company for Sanger sequencing for species identification. Unfortunately, an overgrowth of mold heavily obscured the results in the pilot study. Plates such as the one shown on this slide were classified as inconclusive due to our inability to definitively determine whether NTM was or was not present. In the pilot study, 82% of the plates were classified as inconclusive. Despite this, House 1 showed promising results. You'll remember that I sampled the air pre-shower, then ran the shower for 8 minutes, resampled the air, and labeled post-shower. Then I decreased the humidity and resampled the air labeled post-dehumidification. Following the progression of this enumeration table, no NTM was detected pre-shower. However, 3 NTM colonies were identified post-shower with no colonies identified post-dehumidification. It is important to note that these results were first based on the morphology of the colonies on the plate. As shown on the right, NTM colonies generally present as smaller, opaque colonies and sometimes take the shape of a fried egg. These morphological identification results were then confirmed by genetic sequencing. With these promising results, we slightly adapted sampling protocols to use malachite green, a common antifungal and antimicrobial substance, in hopes that it would help limit mold overgrowth. So as you can see, the malachite green was successful in limiting the growth of mold, and therefore no plates in this second round were classified as inconclusive. Using morphology and colony growth rate, we initially identified 13 NTM-like colonies. All pre-shower and post-dehumidification samples in the 5 houses came out negative. In contrast, 4 out of the 5 houses had at least one NTM-like colony identified on a post-shower sample. To answer my first research question, I looked at the differences between the treatments 1 pre-shower and treatment 2 post-shower. With a p-value of less than 0.05, we concluded that the difference between the pre-shower and post-shower NTM-like colony growth was statistically significant using a Fisher's exact test. In all 5 houses, 100% of the pre-shower samples were NTM negative. On the contrary, for the post-shower treatment, only 20% of the 5 houses were NTM negative, with 80% being NTM-like positive. It is important to note that this statistical significance merely determines that 2 events are not independent of each other and does not provide a causal relationship between the treatments. However, since all controls were tightly regulated and the only change between treatment 1 and treatment 2 was running the shower, this significance indicates that a shower might have played a role in this difference of NTM colony-like growth. Again, we conducted a Fisher's exact test to compare the differences between the post-shower treatment and the post-dehumidifier treatments. Similarly to the first one, in all 5 houses, 100% of the post-dehumidification samples were NTM negative. On the contrary, for the post-shower treatment, only 20% of the 5 houses were NTM negative, with 80% being NTM colony-like positive. Similarly to the first analysis, we found statistical significance when comparing these two treatments with a p-value of less than 0.05. Therefore, the post-shower and post-dehumidifier treatments are not independent of each other. Once again, this does not determine a causal relationship, but considering this study's tight control of outside factors, this significance indicates that by opening up all the windows and using a dehumidifier, it might have decreased the detectable NTM-like colonies in the shower. In an attempt to further confirm our results, we had the 13 NTM-like colonies genetically identified. Unfortunately, the genetic analysis did not confirm the results of our morphological identification. In fact, out of the 13 NTM-like colonies, only 3 were confirmed to be NTM. The genetic analysis results thus nullify the previous results and conclusions, which means that I was not able to conclusively answer either of my research questions. This study had 3 major limitations. First, an extremely small sample size. Second, there is high variability in sampling and working with environmental NTM. Lastly, not all colonies on the plate were picked, which could have led to false negative results. This study has ultimately shown that it is possible to detect NTM in the air of showers, which provides justification for further research on NTM aerosolization. However, future research must include multiple replicates of each shower due to the aforementioned variability in sampling. Additionally, this study shows the importance of using genetic identification rather than the less reliable morphological identification. Although I was not fully able to test the effectiveness of a dehumidifier in reducing NTM aerosols, since there was not enough pre-existing NTM aerosols, this study nevertheless shines a light on the importance of studying NTM intervention protocols. With the rise of NTM pulmonary disease, it is vital that new, cheap, and effective intervention protocols be developed and studied in order to keep all members of our society safe, but especially those who are susceptible to NTM infections. This research process was by far the best learning experience that I have ever had. This year, I learned that research is not linear, but rather a process that can take you down many different paths. In terms of personal development, this research has truly shown me the importance of self-advocacy and for pushing for your own voice to be heard. I also learned that help is always around, you just have to ask for it. For any students who are planning on doing research next year, my only advice would be to truly embrace the process. Cherish the highs and learn from the lows. Welcome the inevitable challenges and never stop asking why. Though this may have been a quote, independent research project, I could not have done this research without the help of so many people. I would like to thank Dr. Honda for all her knowledge of NTM and for being a better mentor than I could have wished for. Thank you to Dr. Chan for her endless support, ideas, and amount of time she spent teaching me in the lab. To Ms. Kobayashi, thank you for all your help on my research, but more importantly, for always being there to cheer me up and to push me to finish. To Mr. Hill, thank you for your help with all things statistics and anything having to do with PCR, because I could not have learned it without you. Thank you especially to my parents for their love and support and for believing in me and funding the second round of my research. Lastly, thank you to my honors independent research class for all the fun memories and support. So here's a list of my sources. Thank you all for attending. I can answer questions now.